Okay, so hello. I am Abby Boyd, and I am doing index cards because I suck at memory. And yeah, I'm just gonna read them off the index cards. And it's my cell project video because you said I wanted to do a video, and I said I'm not writing all that, so I'm gonna do a video instead of writing all that. Okay, so in this video, I'm going to be describing the function and structure of each organelle in my cell project, and I'm doing an animal cell. Okay, so the Golgi apparatus structure. A maze is what some say defines this unique structure. However, it is made of flattened membrane sacs in stacks or clusters. The function, well, that was the structure. Sorry, that was the structure. And the function is, it is involved in transport of lipids announced around the cell and creating the lysosomes and packaging, sorting and modifying proteins for each section of the cell. Okay, that was the Golgi apparatus. Now the chromatin function. The function of chromatin is to efficiently pack DNA into a small place to fit it into the nucleus of a cell and protect the DNA structure and everything in the nucleus and that. Yeah. Okay. Sorry, I'm just it's 10 a.m. To 10 p.m. and I'm all up on coffee, so like that. Chromatin is a complex of DNA, RNA, and protein found in eukaryotic cells. That was the chromatin. Plasma membrane function is primary function is protection of the cell from its surroundings, and it's it lets things in, if it needs in, and it keeps things in if it needs in, and it keeps things out if it needs out. It lets things in, but it's very it's very strict, and you have to be. It's like the VIP list. You have to be on VIP list to get in to the cell. So that was the function. The structure consists of both lipids and proteins. That is what it is, and it is just the outer layer of the cell. It's just it is the protection. It is the protection layer, and it just keeps everything out that shouldn't be out. Okay. Nucleus structure. Found in the eukaryotic cell, it contains most of genetic material of the cell, which is the DNA, basically. Nucleus function. The cell part is only found in eukaryotic cells. It contains almost all of the genetic material of the cell. Nucleolus structure. This is the largest structure in the nucleus, aka site of ribosome biogenesis. Nucleolus function, made of proteins and ribonucleic acids. It combines with proteins after rewriting the ribosomal RNA. Now the mitochondria structure. The mitochondria structure. The mitochondria are shaped perfectly to maximize their productivity, and they're made of two membranes. The outer is like skin basically. And it's it's basically like skin, it's basically a skin to the mitochondria. And the inner layer is folded into little sections. Oh, wait, did I do that? And the mitochondria function is basically the powerhouse of the cell. It pumps energy to the whole entire cell. And it's basically like an alien, Mr. Triplett. You'll love this because it's your favorite organelle, and it's basically like an alien organelle because it's its own thing. It's not it wasn't there all the time. It was brought into a cell at one point. It's not. It's its own thing. It's its own alien. So the flagella. Sorry. The flagella is it's basically like a tail on the end of the cell. Its structure, it just basically looks like the tail on the end of a cell, and it's basically a sensory organelle, and it helps sense things like whiskers on a cat or a dog. That's what I compared it to, and it's a whip-like structure that allows a cell to move. While this is function. It's a whip-like structure that allows a cell to move while all of them are used for locomotion, but they all have a different structure. They're all different structurally for each individual, for an individual cell. Okay, so ribosome function. The cell part makes protein repairing damage or directing chemical process is why this protein is needed. Floating within the cytoplasm or attached to an endoplasmic reticulum is where they can be found. So rough ER has ribosomes on it, and and sometimes as you would say, the hippie ribosomes are found just just floating around in the cytoplasm. So the next one is these are the microtubules. They are the largest cytoskeleton parts in a cell, and they are made of subunits called tubulin. And that was the structure, and the function is basically like conveyor belts inside the cell. They move the things, and like they move the vesicles, granules, organelles, like the mitochondria all around the cell. And there's like they're basically just like conveyor belts. Okay, I'm sorry if this is a little bad. I'm just like hyper and just sorry. Okay, so smooth ER function. Each organelle in the cytoplasm of a cell is responsible for performing certain functions. So this one, yeah. So each of the organelles in the cytoplasm of a cell, yeah, and this one is basically to like protect the nucleus. And the smooth ER structure is not only does it regulate and release calcium, it also ions process toilins. It's described as smooth to tell the difference between rough ER, which has protein synthesis from ribosomes on the surface. This is the microfilaments, and they are like the fine, like twisted fabric basically, and they are like a cytoskeleton. This is the structure, and they're basically just like twisty little. They're not thick like the microtubules. They're thin and they're like twisted, and yeah, and that's the structure. And then the function is they're flexible but very strong and provide a strong support for the cell, as in the cytoskeleton. Okay, so the rough ER function. Ribosomes studded with millions of membranes bound ribosomes help the production, quality control, folding, and dispatch to some proteins. Okay, so structure. Due to having ribosomes attached to its surface, this is called rough sacs called cristae. I know, cristae are formed by the double membrane of the smooth and rough ER. Okay, so this is the cilia. Yeah, cilia. Small hair-like this is the structure of a cilia. Small hair-like structures that extend throughout the cell on the outside of it, in all of the animals, in every animal cell. And the function is they move liquids past the surface of a cell. For single cells, it helps them move. Okay, this is the last one on paper because the other ones I can remember in my brain. So this is the vacuole. It's a membrane-bound sac that carry things in and out of a cell. So it's like it can carry large molecules or small molecules, and it helps things get from like the Golgi apparatus to the nucleus, and from the nucleus to the Golgi apparatus, and from the mitochondria to other places. And this is the vacuole, and yeah, and the lysosomes are, like you would say, are basically like Lysol, and they they like clean up all the destruction and that inside of the cell, and they just like they clean up things. That's what their job is to do, is it's clean up things. And they're just basically little like small like circular little, just like maybe basically sacks of just things that clean up things. and the cytoplasm is the stuff it all floats around in. And it's just basically it's just the stuff that they all, like, sit in and stuff like that. Yeah, okay, so I know that I probably got some of this wrong, and it's going to be counting against my grade, but this was the best I could do. Thank you for listening, and thank you.